Okay, so thank you, Natalia. Thank you for the invitation to present our team and project research. So I will present the laboratory, and Henri will present a project of research. So I am a professor in biomaterials in the team of Jürgen Zipman. So our team is Advanced Drug Delivery System. As you can see on this slide, approximately 50% of clinicians in our team, and we have also some pharmacists, engineers, biologists, chemists. So we have a very interdisciplinary team with many students, postdoc, and master. We also have technical support with some engineers to help us on a biological and analytical analysis. Our team is specialized on advanced drug delivery systems. So what is the concept of a local drug delivery system? If you have a patient who have some disease, local disease, for example, inflammation, infection, or cancer, the patient has to take a huge amount of drug because a small amount of the drug go to the target site. So the consequence is a very fast release of the drug, and in some cases, the concentration is higher than the toxicity level and shortly go under the efficacy level. So that's why we develop some advanced drug delivery system, and the objective is to target the drug at the on-site and have a lower quantity of drug to the patient. In terms of profile of release, we would like to obtain prolonged drug delivery, and the concentration of the drug has to be higher than the efficacy level, but never higher than the toxicity level. So we would like to stay in the therapeutic window as long as possible, depending on the pathology we have to treat. In our lab, all projects start with clinical needs. After the clinical needs, we work to develop a new drug or new medical devices with for drug delivery, and we do all the characterization, biological test, analytical test, and drug release test. As soon as after we characterize the sample, we develop some preclinical tests. We have some model in the lab, so I will show you some of them. And at the end, we can go to the clinical evaluations and go to the patient. In our lab, we have 6 therapeutical fields. We work in otology, in plastic surgery, in gastroenterology, vascular surgery, dental surgery, and maxillofacial surgery. And for each field, we have a team of researchers and clinicians who work to develop the system. In each case, we try to elucidate the mechanism of release. We develop new tools for the product development, and we develop new drugs and medical devices in order to increase the efficacy and limit the adverse effects thanks to the drug delivery, the local drug delivery. So I will focus all the presentation of the lab with medical devices and show you all characterizations that we can do in vitro and in vivo on each case. So in our lab, we modify some medical devices, for example, polyester vascular prosthesis, hydroxyapatite bone substitute, wound dressing, polyester meshes, metal stents, and hip prosthesis. Because in some of cases, after implantation, there are some complications. So in many cases, you have infection in case of vascular prosthesis, in case of wound dressing or hip prosthesis. In case of stents, you have 2 complications, thrombosis and restenosis. And in case of meshes, you have adhesion of the plate and cause some pain to the patient. So for each medical devices, we develop a drug delivery system. The drug delivery system we use in lab for medical devices is the grafting of cyclodextrin. So cyclodextrin is a natural molecule. So the origin is starch. This is a cyclic oligomer, and this cyclodextrin has some glucose unit, conic shape, in order to entrap some drugs. Cyclodextrin has hydrophobic cavity, so some hydrophobic drug can be entrapped and after release after implantation in a patient. We have different kind of cyclodextrin, so we can choose depending the size of the cyclodextrin. You have the alpha, beta, and gamma cyclodextrin, but also some modified cyclodextrin, for example, the hydroxypropyl cyclodextrin, which is the cyclodextrin that we use in the lab. In order to modify the medical devices with cyclodextrin, we work with a team in Lille U1008, and we develop a system to modify the surface of medical devices with cyclodextrin with a reaction of polyesterification. So we use a carboxylic acid, and in this case it's citric acid, and cyclodextrin with many glucose units. You can create a first esterification, and the reaction continue in order to create a polymer of cyclodextrin. The polymer of cyclodextrin is a hyperbranch and cross-link network. We can have a soluble and insoluble polymer of cyclodextrin, and this polymer can interact with a drug by hydrophobic interaction in the cavity of cyclodextrin, or with electrostatic interaction with the carboxylate group of the polymer. And we develop a process to fix the polymer of cyclodextrin on fiber in this example, but in many medical devices, as you can see on this slide. We start with vascular prosthesis. We develop an antibacterial vascular prosthesis, and we work with a company, and we have clinical studies with 40 patients. We develop the same with Pierre Fabre and develop membrane antibacterial membrane with chlorhexidine. With Curazan, we develop a bone substitute for the treatment of cancer in animals. With Bone and Joint Research, we develop a hip prosthesis with cyclodextrin to obtain an antibacterial hip prosthesis in case of non-cemented hip prosthesis. And more recently, we develop a mask with the company BioSerenity, and we have two masks on the market. These masks are antiviral masks in case of COVID-19. And the project with Cousin Surgery, we develop meshes with anti-inflammatory and anti-pain, and we have also a clinical study. Just to show you this project briefly, the problem after a mesh implantation is the pain for the patient, and due to this pain, we develop the system to modify the textile with cyclodextrin. So the process is very easy. It's a common process for textiles. We impregnate the textile in a cyclodextrin. After a curing process and a washing process, we obtain a modified meshes. You can see by SEM the coating of cyclodextrin on the fiber and between the fiber of the textile. In our lab, we evaluate the drug loading. So the drug that we choose is ropivacaine in order to limit the pain for the patient. So we evaluate all parameters as concentration and as time of impregnation, but also the amount of cyclodextrin of the textile. So after evaluate all parameters, we choose the good parameter to obtain the highest amount of ropivacaine on the meshes. After that, we perform some drug release. So in this case, we use a paddle system in order to evaluate the release of the drug. So we have this system in PBS, and at each time we remove a part of the medium and we analyze the medium, and you can see the profile release that we can obtain with two concentrations of ropivacaine. This is the two concentration available in a clinical hospital, because we would like that the surgeon impregnate the meshes just before implantation to the patient. We develop animal models because we have to prove biocompatibility, but also efficacy of the meshes impregnated with ropivacaine. So for that, we have a rat model. So this is a model of colorectal distension. Briefly, we implant the meshes on the abdominal part of the rat, and each day, some rats are tested and we measure the pain threshold. You can see the result on this picture with the black curve is the control, so it's the meshes without anything. And the green curve is the meshes with cyclodextrin and ropivacaine. And you can clearly see that we can reduce the pain to the rat after implantation of these meshes. So the company Cousin Surgery developed a system in order to implement in hospital meshes with ropivacaine, and the clinical evaluation started 4 years ago with 290 patients with two groups. We have a group of cyclomesh, meshes with cyclodextrin and a placebo, and cyclomesh with ropivacaine. And for now, the clinical studies is finished, but we didn't observe for now any differences, any significant differences between the two groups of our tested materials. As I show you at the beginning of the presentation, the polymer of cyclodextrin can be obtained in powder. So we use the powder, and the powder is a polyelectrolyte. It's negatively charged, and we evaluate with BIOMAT the possibility to mix with a positive polyelectrolyte as the chitosan. So we can create multi-layer system, nanofibers, and hydrogel, and I will show you some part of this concept. For the first one, for the multi-layer system, we work on wound dressing, and we would like to treat a diabetic foot infection. The objective is double. First, we would like to add antibacterial compound which use silver, but we would like to limit the diffusion of silver in the wound. And the second drug is ibuprofen in order to limit the inflammation of the pain of the wound part. Sorry. So for that, we use a wound dressing, a polyester wound dressing. We first modify the polyester with cyclodextrin, as I showed you just before with meshes. And thanks to the anionic function of polymer cyclodextrin, we fix silver. In order to limit the diffusion of silver, we apply a multi-layer system composed by polymer cyclodextrin and chitosan. And at the end, we incorporate ibuprofen inside the cavity of polymer cyclodextrin to obtain a double charge wound dressing. On this picture, we can see the first layer with polymer of cyclodextrin and the second layer with silver. We can clearly see the change of color, proves that we have silver and nanoparticles of silver on the surface of the wound dressing. We can build our multi-layer system, and after 21 layer, we can see an increase of the weight of the wound dressing and prove that we have a good multi-layer system. We evaluate the drug release. You can see the first one is release of ibuprofen. After 1 day, we have released the whole quantity of ibuprofen. So this is the target that we would like to obtain to limit the inflammatory response. And about the silver, you can see a very slow release of the silver. And I didn't write the percentage, but we release only 3% of silver that contained in the wound dressing. So this is very nice because we would like to keep silver inside the wound dressing. We evaluate the antibacterial activity of the system, and we prove, compared to a producer of the market, which is Ergotul in a black line, we can see that our two systems, silver wound dressing with silver and wound dressing with silver plus ibuprofen, that we have a better activity of our system. Now we just received from INSERM a new machine in order to evaluate the drug diffusion of our wound dressing. This is the France cells, so you can see this system. We have our wound dressing here, and we can measure the diffusion of the drug inside the medium, and we have different part to evaluate the concentration at each time point. So this is the next step for the measurement of drug diffusion. We also work in an INTERREG project, and we didn't use animal models to evaluate the efficacy of the wound dressing. We use skin pork, and after remove the skin pork, we use this this skin and we prepare some sample to infect the sample, and after this we can evaluate the efficacy of our system, hydrogel or wound dressing, and this is better to evaluate ex vivo the efficacy of this kind of medical devices. For hydrogel and sponge, we work with the maxillofacial surgery services, and we work on the bone replacement, and the problem is the low vascularization of this part for large bone defect. So for this, we develop an hydrogel with the same compound, polymer of cyclohexyl and chitosan, and we can easily prepare an hydrogel, and after lyophilization, we obtain a sponge. The sponge can be cured or not. If we cure the sponge, we have covalent bond between chitosan and polymer cyclohexyl. If not, we have a unique bond. So it's very easy to obtain hydrogel, but Henri will show you in details just after the preparation of hydrogel. In this case, we lyophilize and obtain sponge. We can evaluate in lab the cytocompatibility of this sponge. We evaluate the interaction with osteoblast cells. As you can see on SEM, we have a good adhesion of osteoblast on a sponge, and with the live dead test, you can see an excellent survival rate of osteoblast cells. We did exactly the same with endothelial cells, and we obtained the same result with a good adhesion and good survival of endothelial cells. In order to improve this result, we load the sponge with a VEGF, and we evaluate the profile of release of VEGF. In this case, we have a small amount of medical devices, so we put in a multi-well plate, and at a different time, we determine the amount of VEGF that we can load and release. So you can see on this figure that more the quantity of VEGF at the beginning is important, more you have, more the release is fast. In order to evaluate medical devices or biomaterials for bone tissue engineering, my colleague Feng Devan developed in INSERM project two models, mandibular bone defect model and calvaria bone defect model, and in the same animals, she can do the two defects in the same time, and after implantation and analysis with microCT, she can observe the bone reconstruction. Finally, with nanofibers, we can develop some nanofibers with the two compounds, chitosan and polymer of cyclohexyl, in vascular surgery. In this case, we would like to coat the stent with nanofiber membranes in order to have a first layer with anti-thrombotic activity to avoid thrombosis, and a second layer closer to the arteries to release antiproliferative agent. So we success to coat the stent with electrospun membrane, and we can control the thickness of the membrane with the time of electrospinning, and we can multiply by 3 the thickness of the membrane when the time is increased by 6. We also evaluate the drug release. In this case, it's for vascular, so we adapt the model of release. In this case, we use flow through cells, so it's a dynamic system, and we have a closed system, and we can put the stent inside the machine, and we can coat around the stent agar gels and evaluate the profile of release inside the stent online. So the result shows that we can control the drug release with 2 parameters, the amount of cyclohexyl and the thickness of the membrane. More we have cyclohexyl, more we plunge the drug release, more the thickness is important, more the plunge, more the drug is plunged. And to evaluate the efficacy of our system, we develop animal models. In this case, we choose a rabbit, and we modify the model with a specific jet in order to have a hypercholesterolemic rabbit models. And after some weeks, we do an injury with a balloon in order to provoke a stenosis. And after, we implant our stent to evaluate the potential of anti-restenosis effect. So as you can see in this picture, for example, for a biometal stent, the commercial stent without a drug, you have a score of 1. A score of 1 is a high score, so it means a very important restenosis. And when we have a drug-eluting stent, we divide by 2 the restenosis, and we have a good penetration or good efficacy of the stent. So we can also evaluate these materials with this model. More recently, we developed 3D printing platforms. So with Tech Santé, which is a program between region and country, and also Interact project. And this platform is led by Professor Romanico, Jean-François Witz, and Marion de Hurtevant, and also Vincent Martin and Mathilde Maillard. And you can see all the equipment that we have on the platform. We can print a different kind of polymer, different ceramic metals. We can also do some training for a student and many characterization for the device. We also have a biological lab in order to do bioprinting. So to conclude about our lab, in terms of processing, we can create textiles by nanofibers. We can functionalize all textiles. We can do some hydrogel and sponge. We can do 3D bioprinting and 3D printing, but also I didn't show you all, but we can prepare some nano and microparticles, but also pellets for drug release. In terms of in vitro testing, we have all kind of characterization for materials, but many machines to evaluate the drug release adapted to the medical devices or adapted to the pathology, and in vitro biological evaluation according to the standards. We start to develop ex vivo model as I show you with the pork, and do some histological analysis. In terms of preclinical evaluation, in each case we develop a different model. So I show you 3 models, but we have a list of models that we develop in each case, and Henri will show you just after another model that we develop for the osteoarticular disease. Thank you for your attention. If you have some question, I think all questions are after.
Yeah, I think we will let the questions for after, maybe. They can start already writing in the chat if they want. Thank you, Professor Nicolas, for your presentation. And just one thing, I forgot to tell you before that now we are on YouTube too. So from now, some the webinars can be recorded and then retransmitted in our YouTube channel. So if someone lost the webinar, don't worry, you can watch it in some other moment. Thank you everybody for your participation today. Thank you for the speakers, and don't forget, 12 December we have BioMat, and in May next year we have the BioMat Congress. And so thank you everybody. Have a good day.
Thank you very much. Bye bye.
Bye bye.